Good afternoon, everyone. Welcome back. We're going to start our Wildlife Can't Read the Map quick rapid chats talks so that everyone's going to have 5 minutes, all of our speakers, and we'll have about a minute for transition. And I ask that all of you, if you have questions, please put your questions in the Q&A so that we can hold on to those. So we'll have some time at the end for those questions. Can somebody give me a thumbs up if you guys can hear me, because I am speaking to...
Sound great.
Awesome. Thank you. So I'd like to welcome as our first speaker, Dr. Chris Ray from the Institute of Bird Populations, who's going to talk about his work across the North Coast and Cascades Network. Welcome, Chris.
Hello, can you hear me?
Yes.
Okay, and so I'm trying to share my screen.
Oh,
Okay, I think you can all see it now. All right, so what I'd like to talk about today is what do the birds say about warmer and drier conditions in our NCCN parks? So I've been working with a lot of colleagues from the Institute of Bird Populations and the North Coast and Cascades Network for a while in trying to understand what we can learn from the bird point count data that has been done in these parks. And specifically, IBP has been going into 5 of the parks since 2005, annually surveying birds in the 3 large mountain parks and 2 small lowland parks at hundreds of points every year. And these surveys give us data on many different species. We can determine from that what's been going on. Are the populations increasing in terms of their density at these locations and across the parks? And we can also address questions like, are they shifting in their distribution as time goes on? So after doing a lot of modeling that accounts for the detectability of these birds and other kinds of changes in the climate and many things that we can do, we're able to estimate the density of these birds. And we focus on a species by species type of model. So here we're seeing results from one of our models of the sooty grouse, looking at the density on the y-axis by year on the x-axis of this particular species. So populations in the 3 different large mountain parks, Mount Rainier, North Cascades, and Olympic, are indicated by different symbols and colors here on this plot. And we see that the sooty grouse is quite stable over time in these parks. There are more fluctuations from year to year in the density of this species than fluctuations over time, longer periods of time. So we have some, really interesting changes in density, but no significant decline or increase in these birds across time in these parks. It turns out that the fluctuations in density tend to be explained by climate. In general, the warmer and drier conditions have been better for birds in these parks. So if we look at some other species, here's 3 more species that we have modeled: northern flicker, Clark's nutcracker, dark-eyed junco. The same way, we're looking at density over time. And in general, the density of most birds has increased since 2005 in these parks. Here we were just looking at mountain parks, and we are going to look at a bunch more species and across. And these are combining where possible results from all three mountain parks. We've been able to model about 60 species over time. I'm not going to show all of them in this little talk, but in general, what you can get from these plots is that although there's fluctuation over time, these species tend to be stable in these parks. Here, we're looking at some results from the two lowland parks, and we see a simplified view of what's going on there, where the little red lines, curves, they indicate that birds are increasing at these parks over time, almost uniformly. They're certainly stable in these lowland parks as well. So that's interesting, because we thought maybe the birds would be stable in the larger mountain parks because they have differences in elevation, and so different microclimates that the birds could use if possible. So what about the distribution of these birds? Are they shifting their distributions to take advantage of different climates? So we just hot off the presses have some results on that. And in general, what we see is, yes, we see some changes in distribution. And for, say, the western flycatcher, those changes look like what we might imagine in large mountain parks. The mean elevation at which we find this species has been rising over time, and significantly so in a couple of the parks. But for other species, the situation is more complex. They might be doing different things in different parks. Some significant changes, some not, some up, some down. Here we have mountain chickadee looking like it's maybe going to be going up in these parks, and then western wood pewee really its mean elevation is declining quite dramatically. So we have a lot of complexity here. And one thing that comes through from the results that we have so far is that the most consistent changes are in Olympic National Park relative to the other parks. In other words, in that park, most species are responding to something over time that is causing them to change. I think I'm
Thanks,
about
Dr.
done.
Ray. I think you're done. Yeah, thank you so much. Everybody, remember, if you have questions, put them in the Q&A. And for speakers, I will pop on when you have about 1 minute left. So if you see me, that means if I turn my video back on, you're running out of time. I'd like to introduce Dr. Cathleen Balantic, also doing presenting some bird work. Dr. Kathleen, you're on.
Yes, hello. And I'm just gonna real quick start the presentation and go ahead and swap those views. Give me a shout if anything here looks wrong. Otherwise, I'll.
Looks good.
All right. So hi everybody. My name is Cathleen Balantic, and I'm the bioacoustic specialist at the NPS Natural Sounds and Night Skies Division. So I'm based physically in Fort Collins, Colorado, but today I'll be speaking to you about a collaboration between my division and Lewis and Clark National Historical Park, which I would like to transport you to right now. So here we're looking at a photo of Fort Clatsop, where Meriwether Lewis and William Clark's Corps of Discovery Expedition camped in the winter of 1805 to 1806. And the park is located near the mouth of the Columbia River by Astoria, Oregon. And against this compelling historical backdrop, this park and this region, of course, are home to many bird species that either live here year-round or migrate here to breed in the spring and summer. And if we zoom out on our little map here, sorry, I'm going to move. This is distracting me. So some of these species migrate all the way up from wintering grounds in central or south breed in the park. And regardless of whether they live here year-round or migrate long distances, these species all have something in common, which hopefully you're hearing. They sing to attract mates and defend territory. So many bird species in the park are identifiable to species based on their vocalizations, which, if this is working properly, you're hopefully hearing that in the background. And we've known things about the birds that live and breed here for a long time. In February of 1806, Meriwether Lewis wrote in his journal that yellow and brown flycatch has returned. It is a very small bird with a tail as long proportionably as a sparrow. And he was probably talking about this bird, the western flycatcher. And his journals also pretty clearly make reference to the Pacific wren, this little brown bird that lives year-round in the region. The birds of this area were known to the Clatsop indigenous people long before Lewis and Clark's written records. So suffice to say, people have been observing the seasonal rhythms of these birds for a long time. And as we just heard, things might be changing. According to data from the NPS Climate Change Response Program, since 1970, the park has warmed at a rate of 2.3 degrees Fahrenheit per century. And climate suitability is projected to decline for some birds in the park and increase for others. But at this point, the impacts are complex and not fully known, which allows us to ask the interesting question of how might soundscapes be changing at Lewis and Clark National Historical Park? And we can take audio recordings of the environment to try to answer that question. And that's exactly what the park has been doing. They've been collecting audio data from March through August at 2 sites for several years now. So there's a forested site here in this picture and a wetland site. And the park has collected thousands of hours of audio. It's way too much to listen to by ear. So we use a mix of expert verifications and an automated detection program called BirdNet to process the data. Oh, I only have 3 minutes. Okay. These data are allowing us to start observing trends for different species. And in these plots on the left, we can see how many vocalizations are detected each year. And on the right, we are summarizing the trends across years. So we can start asking more interesting questions, such as when is activity starting, peaking, and ending? Is variation driven by meteorological variables? And do species respond differently based on how far they migrate? We can ask other interesting questions too, such as, so we know that the nights have gotten warmer at the park as well, so we can observe when different species begin singing in the morning and ask whether there's an interaction between warmer nights and sunrise cues. Here we can see the date on the x-axis and the time of day that species are singing on the y-axis. And you can see that these two species often get started singing at dawn, but sometimes earlier. It would be interesting if we could figure out why. We don't know the answers to all these questions yet, but the park is well positioned to start finding them. And I want to put this work in the broader context that wildlife can't read the map. Audio data are being collected at other parks and public lands along the Pacific Flyway. And there's complementary research or collaboration with other government entities, NGOs, and universities. So we're starting to accumulate a lot of audio data that helps us learn more about birds and other animals. And Lewis and Clark National Historical Park is part of that story. So really excited to keep learning from this data set. And huge thank you to Kayla Fermin and Carla Cole at the park. And reach out if you have any questions. Thanks.
Thank you so much. Appreciate that. Again, put your questions in the Q&A. A little time. We're going to switch to other flying things, bats. So I'm going to introduce Katy Goodwin, who will talk about bats. Katy, you are up.
Okay, let me just get my screen going here.
We can see it.
Okay, very good. So I'm a biologist with the U.S. Geological Survey at the Olympic Field Station. Today I'm talking to you about our bat survey work. And this is a collaboration between the USGS, the NPS, and the Northwest Bat Hub. So our goal is to improve understanding of bat ecology in Washington's 3 large national parks, Olympic, Mount Rainier, and North Cascades. There are not very many prior studies of bats at these parks, and the data that we do have are about 20 years old. And then in addition to that, bat populations are under stress due to habitat loss, changing weather and climate conditions, and the fungal disease white nose syndrome, which Michael will cover next. So our study provides critical updated baseline data to better understand the impacts of these stressors on the bats. Our research questions ask what factors are impacting bat activity. So we looked at daily weather conditions, specifically temperature and precipitation. Then we also looked at how time of year and elevation may impact bat activity and how all these factors might interact. Although our study also included Mount Rainier and North Cascades, I'm just focusing on Olympic National Park for today. This map shows Western Washington with the star indicating Olympic National Park. And within the park, we set up 7 elevational transects, 7 transects along elevational gradients. And we sampled each site there multiple times between June and October in 2019 and 2020. We collected our data using acoustic detectors, and which record the bat's echolocation calls. This is an ultrasonic microphone set up here. Then we use a software program to classify each recording to species. These are spectrograms of bat calls in the software. And then for this analysis, we split our data into 2 groups based on what frequency the bats echolocate at. So we have the high frequency group, which is 6 small bodied species, and the low frequency group, which is 4 low species. This is a California myotis at the top and a hoary bat at the bottom. So what I'm presenting today includes a data set of over 11,000 recordings, more than 80% of which were the high frequency bats. The most common high frequency species were California and Western long-eared myotis, and the most common low frequency were silver-haired and hoary bat, with up to 9 species total per transect. When we ran our models, we found that the single variable most strongly related to bat activity for both groups was the time of year, and we saw bat activity peaking in early to mid-August. Here in the next few slides, I'm showing model predicted bat activity in relation to time of year, which is on the x-axis. The activity is on the y-axis. And then in combination with the other factors, temperature, precip, and elevation. For temperature here, we have the warmest temperatures in yellow and the coldest in purple. So we see a positive effective temperature for both groups. And then for precipitation, 0 daily precipitation was in yellow and the highest precipitation is in purple. Again, both groups had a similar pattern, and here we have a negative effect of precipitation, so lower activity when there's more precip. And just for both temperature and precipitation, we saw a stronger effect for the high frequency bats, and we think this is because they are smaller bodied, so maybe less tolerant to those inclement weather conditions. And then for elevation, we had a more complicated pattern here. For the high frequency bats, the activity was higher at lower elevations during the summer, but then around mid-August that switches and becomes higher activity at the higher elevations, which is interesting. This time of year in mid-August is when the bats are finishing rearing their pups and moving to fall swarming. So this suggests that they're using different elevations for different parts of their life cycle. The low frequency bats, however, showed a more consistent pattern with higher activity at that higher elevation throughout the season. So just quick to sum up here, we saw positive effects of temperature and negative effects of precipitation for both species groups. Both species groups had a strong seasonal pattern, and then elevation was a little more complicated. So our upcoming work that we'd like to do next includes incorporating some habitat variables, looking at individual species patterns, and then looking at other metrics like occupancy modeling and species richness. So that's all I have today. Thank you for all those who were involved.
Thanks for that, Katie. Again, reminder to put questions in the Q&A. We should have about 10 minutes at the end. I will next introduce Michael Hansen, also speaking to us about bats. Michael, you are up.
Let me unmute
I
myself.
can see your screen.
I'm working on it. Hang on. Okay, my name's Michael Hansen. I'm with the U.S. Geological Survey, and I'm here to talk to you about white nose syndrome disease surveillance. So white nose syndrome is a disease of hibernating bats. It's caused by a fungus, Pseudogymnoascus destructans, or PD for short. And so this infects bats during hibernation and causes them to wake up and burn through their winter fat reserves. The disease was first discovered in 2006 in New York, and it spread rapidly across the East Coast, killing millions of bats. And then in 2016, it unexpectedly jumped all the way over to Washington State. So in response, the National Park Service developed a regional white nose response strategy to quickly implement bat population monitoring and disease surveillance across the region. And this was done in collaboration with federal, state, and university partners. And it also included acoustic monitoring and experimental work. But for today, I'm just going to talk about the disease surveillance aspect. So disease surveillance for bats in the West is just a little bit tricky because we don't actually know where bats over here hibernate for the most part, but they generally don't use the caves and mines that you see Eastern populations using. And so hibernacula, which is what winter roosts are called in the West, the ones that have in these little tiny crevices. So even if we did know where more of them were, it's not a space that we could just walk into. So we don't have the opportunity to go inside hibernacula and observe bats while they're hibernating and actively being infected, and also where most of the mortalities occur. So instead, what we do is we capture bats as early as possible in the spring after they've left their hibernacula, but before they've fully healed from infection or groomed away all the evidence of the fungus. And we collect samples by swabbing their skin. We also swab the surfaces where they roost and collect their guano. And then these samples are tested for the presence of pDNA, so they can tell us if the fungus is present, but they can't tell us if bats in the area are actually affected by the disease white nose syndrome. We can look for evidence of the disease by examining bats for scarring on their wings. This shows a healthy and a post-white nose bat wing. And then we can also look for a byproduct of the immune response to fungal infections that fluoresces orange under UV light. But to confirm the presence of white nose itself, we have to collect a tissue sample from live bats. And so we do that using this little 3 millimeter like hole punch, basically. And we can also submit any carcasses that we find in the winter or spring for histopathology. And that looks for these diagnostic lesions and fungal material inside the bat's skin. So this map shows all 36 across the network where we have sampled since 2017. Some of these have been sampled once, some have been done annually, but basically we've collected a lot of samples. And this map shows the current status of all of those sampling locations. So all the gray dots have never had a positive sample. All the half circles have PD positive, so that means we have genetic evidence of the fungus. And then the red circles, the fully red circles, are white nose positive, meaning we have both the genetic evidence of the fungus paired with symptomatic disease confirmed from tissues. And this table shows the sampling history from all 6 positive sites, as well as some of our more consistently monitored sites from across the network. And we first detected PD at Mount Rainier during that first year of widespread sampling in 2017, but just at that one location. So despite sampling annually across a bunch of different sites each spring, we had no detections for 5 years. And since 2021, new detections have really accelerated, and we've seen white nose symptoms and some mortality at 4 sites across Mount Rainier. The progression of disease has been different at different sites. You can see how slowly it progressed at site number 1, where PD was detected 7 years before we saw any symptoms of white nose syndrome, compared to site number 4, where it was consistently negative, but we saw sick and dead bats immediately the first year that it ever tested positive. And we didn't have any detections at any other park units until this last spring, when guano samples from Olympic and North Cascades both tested positive. So we're hoping to capture bats at those locations to look for symptoms of the disease. And these detections make sense in the context of sampling across Washington. We had that first detection just north of Mount Rainier, and that sort of radiated out to overlap some of these other units. And it's been a little bit more slow here than on the East Coast. You can see on the bottom right how much farther the disease had spread in the initial years of the outbreak over there. We've also had fewer mortalities, so it's difficult to pinpoint why exactly this has progressed differently, but it could be some of those differences in hibernation ecology, and also that we can't really observe it in the same way. All right, that's about it. Thanks so much.
Thank you, Michael. I would ask that speakers, if they can remember and are able to turn on their cameras while they're speaking. Again, put questions in the Q&A, and we'll have some time for that at the end. I'd like to introduce our next speaker, Dr. Arjan Meddens. I hope I pronounced that right. Who's going to talk to us a little bit more about rock habitat. Welcome.
Yeah, thank you. I'll try to share my screen here. And then let's see how this goes.
Yeah, we can see it.
Does this look good?
Looks great.
Great. All right, well, thanks for organizing this. And I'm excited to tell you a little bit about our project. This is a NASA-funded project, and we started about a little bit over a year ago. And our goals here are to map and protect rocky habitat in the Cascades Mountains for conservation of climate-sensitive species. So we're thinking also about these climate refugia and such. So I'll just give you a really quick overview of this project. So for the motivation for our work is like we have all this broken rock habitat, and they're really essential to survival of many species, such as the bats we just heard about. And there is some data, but the data is fairly limited in scale and scope and level of detail if you start looking into the available data on rocky habitat. And so what we want to do with our partners from the state and federal agencies is to characterize these broken rock habitat to improve basically the monitoring of species and mapping of the species, to plan maybe future conservation areas, and then also to inform day-to-day management activities. Okay, and so here, this is sort of the plan that we laid out in our proposal. So we're looking at sort of the Washington Cascade Range is sort of our study area. And so our objectives are to first sort of detect any rocky land cover that we can detect, and then go into specific field sites where we do some drone and field sampling, and then really look at different metrics for these rocky habitats. So think about like surface roughness and the depth of the talus and maybe rock sizes. And as we like sort of develop these data layers, we want to then develop these species occupancy models for 3 of our focal species, which include the American pika, the hoary marmot, and the large mountain salamander. And then we want to sort of map these or build occupancy models for these focal species. And then in our last objective, to develop maps and data for sort of management of these areas and these rock areas. Okay, so then I'm just going to quickly sort of go over 3 sort of papers or products that we're sort of have been working on and are almost sort of, well, they're in various stages of being submitted or published. So the first one is sort of a review paper where we just sort of talk about rocks as refugia. So where we sort of lay out sort of all the novel methods that we could use to characterize rocky habitat. And then we're thinking about these above ground characteristics, so on the surface, but then also the below ground like surface, like the crevices and pore size and the substrate depth as well. And then, yeah, obviously sort of the surface ones where we think about rock shape and even distance. And these metrics can then sort of be translated in some sort of like habitat characteristics. But obviously these will be dependent on what species you're interested in. So, that might depend a little bit on what you're looking at there. And then sort of we then link then some of these things, like how we then can sort of do the how we link that with data. So that could be satellite data or drone data, and et cetera, et cetera. Let me see here. Good. So then the second progress is where we're just mapping the the rocky land cover across, like across the Cascade range. And so we've built a Google Earth engine algorithm where we just sort of basically classify whether it's rocky or not. And you can see that here in the B screen, sort of like if it's rocky land cover, we classify it as present or absent. We also have sort of a finer resolution model. This is from NAPE imagery that we have for a select areas where we are maybe more interested. But basically for the whole region, we have this here on the top right. And then just checking, I couldn't see if am I out of time or I have a minute left?
You're very close. Yeah, you're pretty much at the end.
So real quick, so then we used also a base method where we used iPads to do a structure for motion to like characterize these rocky talus slopes, and then use a sort of a segment evergrain, which is sort of a computer program that segments every single rock, and we're building models to characterize these at a really fine resolution. And then the next steps are to scale this all up, build species occupancy and distribution models. And then we were working on a citizen science project for pika data collection as well in Mount Rainier National Park. And then finally, we want to really get to the implementation of these data layers into state and federal management and monitoring plans. So thanks, and I'll leave it with that.
Thank you very much. Again, reminder to put your questions in the Q&A if you have some later. And I'm going to ask Dr. Laura Prugh to come on. I see her camera's on already. Welcome.
Whoops. Okay, wait. Are you seeing my presentation? Let's see. Slideshow.
Yes, we see it.
Okay. Great. Well, thank you. I'm Laura Prugh, professor at University of Washington, and I'm going to be presenting some of the key findings of my former master's student, Kayla Shively's work, looking at diet and partitioning among mesocarnivores in the North Cascades associated with the Fisher reintroductions. So this is a release of fissures of a Fisher release in the South Cascades in Mount Rainier National Park, which I was fortunate enough to attend. That's me and my daughter in the upper left corner of the photo there. And so fissures were released in the South Cascades starting in 2015, and then in the North Cascades starting in 2018, I believe. One factor, fissures are one of the most commonly reintroduced carnivores, and quality is likely quite important to their success of reintroduced populations. And in particular, having sufficient prey of the optimal size, which for fishers is medium-sized mammals, or so snowshoe hares are pretty ideal. Porcupines, mountain beavers, so kind of large rodents, and lagomorphs are ideal prey. They can consume a wide variety of prey types, but the smaller prey are more energetically expensive for them to forage. And when fissures are returned to the landscape, it may be quite different from when they historically occurred here. And in particular, the abundance of some of the dominant competitors like coyotes and bobcats might actually be higher now than it was historically. So we wanted to look at the diets of these mesopredators to see if competition might be playing a role in limiting the fisher recovery in the North Cascades, so that the recovery in the South Cascades population has been doing a bit better. Those fishers have had higher survival and more rapid expansion than the fishers in the North Cascades. So we contracted with Rogue Detection Dog Team to survey and collect carnivore scats of fishers and their competitors in the North Cascades reintroduction area. And we also had scats that had been collected just for fishers in the South Cascades at fisher den for a comparison. And we used fecal DNA metabarcoding to identify the diet items in the scats. So I'll just quickly go through 3 of the key findings from this work. So the first relates to comparing the fisher diets from the North Cascades, where they're struggling a bit, to the South Cascades, where they're doing a lot better. And we found that fishers in the North Cascades are using a lot more of those smaller alternative prey than ones in the South Cascades. So here highlighted in purple are those large, the leopards and mountain beavers that are their preferred prey. And we see a much higher proportion of them in this orange bar here is from the South Cascades compared to the more green bars that are from the North Cascades, where they're consuming more small mammals and small birds. We also saw much higher diversity of prey items in the north, 71 prey species in the North Cascades compared to 22 identified in the south. Ooh, okay, and second, we found that the primary predators of those optimally sized leopards and large rodents were the bobcats and coyotes. They were the main ones consuming those in the North Cascades area. And lastly, we found that the amount of diet overlap between pairs of carnivores related a lot to how similar they were in body size. So the more similar they were in body size, the greater the diet overlap. So here, fishers in the orange paired with the other species had fairly moderate levels of diet overlap. The highest was between bobcats and coyotes. Marten had lower levels of overlap with the other carnivores. So kind of the main takeaway we had from this is that it's certainly possible that is restricting access to optimally sized prey for fishers in the North Cascades and maybe contributing to their slower recovery. So getting some more abundance estimates for these dominant competitors would be an important next step. All right, thank you very much.
Miranda, you're muted.
Thanks, Laura. Sorry about that. Next up, we have John Boetsch. Reminder to turn on your camera and for folks to put questions in the Q&A if they can. And for all panelists to stay on till later for all those questions that are coming in. We can see your slides, John.
Great. Okay. Well, greetings, everyone. My name is John Boetsch, and I'm pleased to talk today about monitoring elk in and around Lewis and Clark National Historical Park. To begin with, I'd first like to acknowledge the tremendous role that others have played in this project, especially Paul Griffin, Carla Cole, Kayla Fermin, and Emily Scott. Their work and contributions are the basis for this presentation. I'd also like to mention that Kayla will be giving a follow-up presentation next on some related work using GPS collars to track elk movement. So why are elk important to Lewis and Clark National Historical Park? For starters, the park enabling legislation emphasizes the importance of preserving and interpreting the historic, cultural, scenic, and natural resources associated with the Lewis and Clark expedition in the winter of 1805 and 1806. And as it turns out, elk were and are a crucial part of that context, as stated in some of the park foundation documents. Reports of abundant elk in the vicinity of the future fort site were one of the determining factors in the vote among expedition members to overwinter there. During their stay, the expedition killed more than 130 elk, which sustained them during their winter encampment and provided them with meat and leather for their homeward journey. The native Roosevelt elk that we see in the Clatsop Plain today have descended from the herds encountered by the expedition. In sum, We value elk as an important natural and cultural resource with both historic and present-day significance, and that's why we have invested in our monitoring programs. So what are we doing and how? First, we conduct pellet surveys at approximately 65 points arranged on a unit-wide grid, clearing the plots in the fall and then returning in late winter to tally pellets and collect additional data to model detectability. Clearing the plots in the fall standardizes the timeline for accumulation of new pellets over the cool wintertime months, which helps to minimize differences in pellet decay rates due to temperature. We also conduct road surveys once a week in alternating months, where 2 observers drive the 4 routes highlighted in this map to record numbers of elk, location, and composition of each group observed. These routes are not random, and unlike our pellet surveys, road surveys do not include methods to account for detectability, so these results cannot be used to infer changes in the underlying number of elk in the area. Instead, the objective of these surveys is to provide an index of elk viewing opportunities for visitors. So what have we learned so far? First, a few important caveats. The results I am presenting are preliminary, given that our analysis is still in progress, and these results do not yet include the most recent years of data. That said, the pellet survey analysis is showing a significant decline in both the relative use and occupied within the Fort Clatsop unit, with a decline of over 2% annually for both statistics. Road survey results have shown broad variability in group size across years, with composition and group size also varying seasonally. And as they say, a picture is worth 1,000 words, and so perhaps a video is worth 1,000 pictures. To highlight the spatial component of shifting pellet deposition over the years, this video shows how raw pellet density has fluctuated at each grid point, and also how these fluctuations seem to correspond in time and space with management activities by the park, including trail updates and tidal restoration efforts. We're attempting to explore these relationships further with the recognition that there are limits to what we can determine with the data that we have. Similarly, for the road survey data, we're seeing elk distribution shifts over time that correspond to restoration efforts inside the park, as well as with development pressure to the north and northwest of the park. We've been working on relating these shifts to data from a related protocol on landscape change, and we'll be including these results in our upcoming report. To give one quick example, this graphic summarizes all years of data for one of these routes to the northwest of Fort Clatsop unit, showing a concentration of gray circles representing historically larger group sightings in a field that is now surrounded by development. To summarize the core takeaways, we're using pellet surveys and driving surveys to track seasonal elk use and viewing opportunities in and around Lewis and Clark's Fort Clatsop unit. Preliminary results indicate distribution shifts and a slight downward trend in area occupied and relative use. These changes seem to correspond in time and space with park management activities and adjacent land use changes. To close, I'd just like to acknowledge again the terrific partnerships and contributors that have made this possible over the years, and thank you for your attention.
Thanks, John. And our last speaker of this session, Kayla Fermin. Kayla, can you turn on your
Yes.
camera? Thank you.
Hi, can you see my screen?
Yeah, I can see you and your
Great.
screen.
Wonderful. Thanks. Hi, everyone. Thank you, John, for the introduction to our elk program. My name is Kayla Fermin. I'm the Natural Resource and GIS Program Manager at Lewis and Clark National Historical Park. Like John mentioned, we have implemented a supplemental monitoring program to gain insight onto elk movement, elk home ranges, and herd fidelity using GPS collars. Sorry, I'm trying to move windows over here. Okay. In 2019, the park worked with state agency partners to deploy 6 GPS collars on adult female elk. These first collars were configured on a 13-hour fixed interval throughout the year and a 7-hour interval during calving season. To understand the greater variation, we deployed additional collars with updated GPS fixing frequency to every hour. So today we're going to follow the journey of 2 females over the last year. I want to preface this with this data and the preliminary data exploration are for individual elk, and these patterns may not always reflect larger patterns of an entire herd. Each of these cows received GPS collars by Oregon Department of Fish and Wildlife, one of our partners, on March 23rd, 2024, outside of the park boundaries, and again, are fixing every hour. So we're going to focus on 82105, or we're going to call her Elk H. After receiving her GPS collar over here, 4 hours later, she moved north into the park where she spent the duration of the day. These points on the map show the locations where the GPS collar provided her location for March to January 29th of this year. We can utilize kernel densities to visualize where these features are concentrated. Now, these are just the locational fixes clustered. And naturally, we want to find patterns and make sense of these clusters. Are these areas random? Is there some underlying pattern? And so to understand those spatial patterns that may be present in the data, we can run spatial statistics tools. So these red features here represent hotspots where those values are clustered, and it is unlikely that the observed pattern is the result of a random process. So there's some other pattern happening here. These areas were in hexagons to reduce sample bias and also to better represent the connectivity and movement in the landscape. This is the same map for Elk H, just zoomed in. And so we're looking at how these patterns can help us understand their behavior and how their behavior impacts the landscape, and reversely, how management of the landscape impacts their behavior. So here at the top of the map is a site named Otter Point, which is one of our restored wetlands within the park. It is likely here that H is moving across the Lewis and Clark River to access this other site to the south. And this behavior has been observed before. So here's a picture of the group of elk crossing the Lewis and Clark River at that exact same spot on February 13th, 2023. We can continue to visualize these patterns to understand how their behavior changes. So we're looking at a different elk, Elk K, likely also using Otter Point regularly to cross the river and head down to this southern portion. This is a proposed restoration project called East Atoll. Also, additionally, LK is using Colwart Creek, which is a restored wetland. So all of this, what does it mean for management? As we plan future projects, we can use this data to inform the design, whether that means restoring that, ensuring that there's plantings to provide that forage in the winter months where they're primarily using these sites in the winter, or we're protecting plantings from the browse as those plants become established. And additionally, we can use this data to address challenges and develop creative solutions for human-wildlife conflict. Here we're looking at Elk L, who was collared on the West Highway 101. So this map is very chaotic intentionally. It's showing all of her tracks along the highway and all around over the last year by month. And so the blue dots along Highway 101 are generated based off of every time she intersects that road. And if we look at by month when she's intersecting the road, this is happening during the months of August to March, with the highest crossings happening in December and January. And so this data presents, thank you, this data presents further questions. What are additional barriers to elk movement? Is it roads? Are there elk herds that are more likely to cross roads? Obviously, the Lewis and Clark River is not a barrier. Is it a barrier to some herds? Are there certain times of year that there's more frequent crossings? Are there hotspots of those crossings? How can we safely live with elk in this landscape and understand that we both occupy the same place? So this data exploration is only 10 months of data. And with more data collection and further analysis, we can decipher more patterns. This GPS collar data complements our pellet and driving surveys, serving a greater understanding of local elk ecology and can inform our management decisions directly. And, we are tasked with caring for these special places and beings as stewards of this land. And so we're going to continue to conduct the science, as scientific observation is a fundamental value of the Lewis and Clark expedition and continues to inform the management of the Park Service. Thank you.
Thanks, Kayla. And to all the speakers, if everyone will turn on their camera, we're going to do a Q&A session now. And I also realized I failed to introduce myself. So I'm Miranda Terwilliger. I'm the wildlife program lead here at Olympic National Park. And I've been delighted to hear about everybody's science and results in these parks. First question I have, it looks like it's for Chris. And it looks like, Chris, are the bird results written up somewhere? Should be a quick answer.
No.
Nope.
We're in the process of preparing a manuscript right now.
Awesome. Another one for you. This is from Macy. Do you have any more specifics about bird species decreasing at Olympic National Park, or is it overall they're decreasing due to changes in climate?
Well, I hope that I said increasing in general, because that was what we found. Sometimes I say the opposite. Generally, the birds are increasing, and that is due to similar patterns with the bats, where they prefer drier and warmer conditions, and that's probably why they're increasing right now. I'm not sure if I answered the entire question.
Macy, did that work for you? Is that what you need to know? I think that was sufficient. For our bat folks, a question from Haley. What can we do to prevent the spread of white nose syndrome?
I mean, if you're going to go into underground spaces, it'd be great if you didn't. I mean, you can disinfect, but it's best, honestly, to not wear the same shoes underground in like positive areas as negative areas. But that said, it does kind of look like it's becoming endemic across the West slowly over time. So I don't know how much there is in the long term that we can do.
Another question for you, Michael, one from David. Is there any investigation of white nose syndrome near Mount St. Helens, especially Ape Cave?
I mean, I'm sure that there is. That would be Abby Tobin from Fish and Wildlife. That's sort of her jurisdiction. So I know that there are a couple of areas down there where we've even found myotis using caves in the winter, which is unusual. So I'm sure that they're investigating them, but I don't know any of those results right now.
A different David wants to know whether or not you're communicating with Forest Service folks about white nose syndrome found nearby.
Yeah, all of our test results run through the National Wildlife Health Center and through Washington Department of Fish and Wildlife, and those get uploaded to their publicly available maps with contact information for people if they wanted to get in touch with different folks in their area.
Dr. Prugh, how are you measuring the fisher reintroduction success? It seems detections could be more difficult in the North Cascades.
Yeah, so that's mainly from the work that the reintroduction team has done, like monitoring their survival. So the survival of the reintroduced fishers was about twice as high yeah, I don't want to put an actual number because this is just from memory, but it was substantially higher in the South Cascades than in the North. And then they've recently implemented pretty extensive camera surveys throughout the recovery areas. And there's been more time in the South for expansion, but even beyond that, there's just much greater occupancy in the South than in the North. Yeah, so those are the main ways that that's been assessed.
Another question about bats. Michael, any thoughts as to bat behavior or environmental factors influencing the rate of spread in North America, in Western North America versus Eastern North America? Sorry, I didn't quite translate that.
Yeah, there's a lot of different possible answers to that. Two of the big ones are in the east. Bats tend to come together in those big communal sites, and so they can travel like hundreds of kilometers to get to those, and there can be hundreds or thousands of individuals in each one. And here we seem to have these little tiny ones, and so that's just a couple of individuals. So just if you're thinking about like person-to-person spread of a disease, it's a smaller amount of individuals coming together. And then also just that those sites are usually within about 30 kilometers of their summer ground so far. So like spatially, those bats are just not moving as far. So this is sort of continuation of bats moving across the landscape is just really different here. And then also microclimate, you know, microbial communities. There's a ton of different possible answers to that question.
Great. And then I have one that asked if they can get the email when the Algoby et al. Paper comes out. So I think there was a yes. If you want that, please send an email that's in the comments to Arjan. He's got his email address in there. So if you're interested in that. Trying to see if there's any more questions. Looks like we have a little time. So if folks want to add extra questions.
Well, I have a question, if that's allowed.
Yeah, panelists.
So as
Any
we're
questions?
mapping rocky habitat, I'm very interested in what you could tell me about the beds being in crevices or talus slopes. Is that like, I remember seeing something about like they're more in quarries in the east, but where are they over here is a question, or where are they in the west?
So as far as over winter, in like the Intermountain West, they've definitely found some evidence that they're using talus slopes as hibernacula. And then Karen Blavis up in Alaska found sort of like soil covered talus slopes under forest canopy cover using just like little voids under like tree roots and things. So a lot of really different small crevices. I think I'm working with some of your folks at a Navy base in Washington, and I've even seen some Townsend's long-eared bats coming out of talus slopes that we've surveyed over there. So they're using them during the summer too. And yeah, definitely a possibility for hibernacula. We just don't know quite to what extent.
Yeah, cool.
Any other questions from folks? If not, I just want to thank everybody again for your presentations. It's really great to see all the work that's going on in these parks and how they all help us as managers manage our parks better and our resources. So thanks again. Appreciate it. And I'm going to hand it over to Hazel. Oh, wait, there's one more question that just snuck in. So good. Is the USGS working with private bat control companies to learn more about their local housing?
Private bat control companies. Well, so our project is a Park Service funded project. So like we're pretty specific to Park Service land right now. So all of that would be done by state, probably like WDFW. And I know that they do take information from private folks on where bats are roosting. And I don't know to what extent they're talking to pest control agencies, but I imagine there's some communication there.
And Miranda, it looks like we have a hand up. If that person intended to, they can go ahead and ask.
Can't see that, so go ahead, whoever that is.
All right, we can move ahead. It may have been a mistake. Thank you. Thank you.
Yeah, sorry, I do not see the hands for some reason.
It's my job.
Again, thanks a bunch, and handing it over to Hazel.